Thank you so much, Arjun, and thanks to both you and Shalab for the introduction or the invitation to speak at this amazing conference. It's really just been a fantastic few days for me. Every talk I feel like has some method or biological or theoretical insight that's directly relevant to the work we do. So just kudos for this amazing conference. So what I want to talk today about is development. And we've had a number of talks at this conference about development. And typically, people start off with a slide like this, where in a single cell context, we're really thinking about trying to deconvolve the complexity of this incredibly complicated process, right? So you see a lot of different things happening in these different embryos as they develop. Cells are dividing, they're moving around. There's adopting different sizes. Some of them are turning fluorescent. And they're doing these all at very precise times as well. And so I'm going to tell you a little bit in the beginning about some of our work to map gene expression across development in this organism in the top right corner here, the C. elegans embryo. I'll tell you why we think that's a great model in a second. And then leave a little bit of time at the end to talk a little bit about the temporal control as well about development. So why is C. elegans a great system to apply and sort of develop single cell methods in addition to answer interesting biological questions? We're certainly very interested in the conserved mechanisms. So obviously C. elegans, while it's small and transparent and develops very quickly, it has muscle cells and neurons and all the basic cell types that we don't love from more complex mammalian systems. Perhaps more real critically important, though, is it has this really robust mode of development. And so back in the 80s, John Sulston mapped out this entire pattern of cell divisions that each C. elegans embryo goes through as it's developing from the zygote to ultimately a worm that swims away. And what's unique about this system is it's incredibly robust. And so, we saw a lot of examples of developmental robustness in other organisms throughout this talk. In C. elegans, the actual pattern of cell divisions in every wild-type individual is invariant. And so we can look at any given individual and follow a particular pattern of cell divisions. We know what that cell is going to become in terms of its cell fate. So how do we take advantage of this invariant lineage to map gene expression across the lineage? We've seen, again, in this conference, many examples of the complementary advantages of imaging-based approaches and sequencing-based approaches. So you can see in this movie that's developed, that's going along the left side of the screen here, as the embryo develops, we can really see which cells are dividing and how they're all related to each other. If we follow this movie very carefully, and we could actually follow from the zygote all the way to the terminally differentiating cells. What are the pattern of cell divisions that led to that? And with very high certainty, right? And we can see, some cells maybe will be fluorescent. We get very high temporal resolution from that as well. You can see exactly when particular reporter gene comes on in this case. However, the throughput of that is much lower than in terms of the genome than say sequencing-based approach, where we can do large-scale sequencing. We can get information genome-wide about how different genes are expressed across cells. What's missing in the sequencing case, and the previous talks notwithstanding, amazing approaches that are coming online to try to get around this, but it's basically a static snapshot, right? So each of these dots in this case, it's a UMAP projection of a single cell data set is a single cell at a single time. And we have to infer based on something about the pattern of trajectory, the similarity of cells that form these trajectories of similar states, that there's some lineage relationship there. All right, so I'm going to start with a little bit of the background on the imaging approaches. We basically, our goal when I started my, getting into the system back in the early 2000s now, was to use this as a system where we could really think comprehensively about mapping gene expression, both for every gene as well as in every cell of the organism. And because of this invariant lineage, we can actually make a list of all of the cells of C. elegans at any given stage. And then, if we can figure out the expression of each of those, we have, in principle, a complete description of for example, So using a movie like the one I showed you on the last slide, we can use cell tracking software, the program called Starry Night that was developed by Zhuang Bao and I back when we were postdocs to track the cells across time through cell divisions, quantify gene expressions, noted by these funny yellow basketball-looking object here to account for local background correction. And the output of that is the lineage tree of that particular embryo that we watched the movie for. That's denoted here in this purple color. And we can quantify in each cell at each minute throughout development from these movies the expression of a second color fluorescent reporter. In this case, it's a GFP fused to this transcription factor FA4, which is a conserved master regulator of gut development across all animals. Right. And so this is a nice lineage tree. It shows you, even for presumably most of the audience isn't the expert on the elegans lineage development like I am, but now if I told you that this gene is expressed only in gut cells, you have some sense of where in the lineage the gut comes from. You can see there's several different clones that produce gut cells. Some of them are larger, some of them are smaller. This one on the right here is the intestine. The little small ones are the rectum. The rest of that is sort of the more complicated pharyngeal apparatus that is involved in eating and swallowing food. And so it's a single cell resolution, high temporal resolution map of gene expression for that particular reporter. And the great thing about this, we just turn the crank over and over again for many different reporter genes. And so here's an example of just 4 different genes that are, again, conserved regulators in different tissues: muscle, gut, intestine, gut more broadly, and neuron development. And because it's the same lineage every time, we can say that this cell in this embryo that's a MyoB expressing embryo is the same cell essentially as the cell from this N3 expressing or this cell from this FA4 expressing embryo. And we can map those all onto a single reference lineage now, where we have the expression of these hundreds of genes all mapped to the organism at single cell resolution. And that's in this database, that EPIC database. Expression patterns of serotypes, not a medical record database. Right, so switching gears to sequencing studies, right? This has just been an amazing explosion the last few years in terms of the ability to map gene expression across entire developing embryos and time courses. This is an example from zebrafish that I think we saw in several previous talks. Looking at going from the very earliest uncommitted states in different tissue and germ layers. And the basic principle for these approaches, as probably most of you are familiar, is that you have some different stages of embryos. Maybe you have them stratified in different wells. Maybe they're mixed together. You separate those into individual cells. You run them through something like a 10x or a drop-seq or an indrops, or some other single cell approach, and then you get transcriptomes for them. Obviously, they're very sparse transcriptomes, and the hope is that there'll be enough information there to be able to assign that cell to its appropriate place in the developmental trajectory. And in most organisms, this is a discovery operation, right? Where we actually, we know the basic broad brushstrokes of how cells are committed to different fates. We don't necessarily know all the details. In C. elegans, that's not true. We actually know the right answer for this. So this is a great test case to see how well we can do for this, right? And this is work. There was a great collaboration with Jonathan Packer, who was a grad student in Bob Watterson's lab and co-advised by Cole Trapnell, and Chen Zhu, who's a grad student in Jin Young Kim's lab in our biology department at Penn, right? So I'll just start this manually. There it goes. So we did this. I'm going to go through this atlas relatively quickly because it's published, and just highlight the key methods and a few of the, I think, interesting insights that we got from this approach. This is an atlas of the C. elegans embryo based on about 100,000 cells from all different stages. And this is a 3D UMAP projection of those cells. And they're color-coded by, you can think of that as basically developmental stage. So the green cells are early progenitor cells, and as they go up to the blue and then red different tips of these different branches, that's the different cell fates radiating out and, as the cells develop. And I don't want to dwell on it too much, but basically, it turns out, and then we did a lot of work downstream to validate this, but we can take mixed stage embryos and then figure out after the fact what stage embryo each cell came from by basically mapping the cell's transcriptome to a whole embryo, really high resolution whole embryo time course data. And this on the right is just a validation of that, where if we age the embryos, we lose the early cells, one of several validations that we did. So we know for each cell what time embryo it corresponds to. And obviously, like people typically do in these atlases, we went through and used marker genes to annotate different tissues and cell types, right? So this is looking at the entire data set. You can see all the major tissues, muscle, skin, different types of neurons, intestine, and so forth, as well as more specialized cell types. If you're C. elegans cell type wonk of some kind, comes, oh, I'm really interested in the head mesodermal cell, and I can say, oh yeah, we have the head mesodermal cells. We have basically every specialized cell type, that we can, that it's a very different cell type, we can identify them in these. And this whole organism level view doesn't give you a totally resolved picture of all the cell types. And this is something I think has been hinted at a few times. Just want to emphasize it. You really have to do some sort of iterative analysis to try to piece apart more specialized cell types. So for example, zooming in on this group of cells, which is the ciliated neurons, the sensory neurons, if you reproject those, now you can get a much higher resolution map, where I think we can go through again using literature markers, and each of these terminal branches is a neuron, and we can identify each of those. And basically, this is all of the ciliated neurons that are present in the embryo, which is pretty cool. There's a total of 20 born in embryogenesis. I think there's one that's not on the diagram, it's somewhere else in the plot. So that's great. That's, like I said, we know the right answer, and basically we can identify all the cells we expected, and we don't find any cells that we didn't expect to find, which is also sort of nice. And on top of that, we can start seeing some hint that maybe we can also identify progenitor and lineage relationships. And so, like in this area that has this mark here with these black hash marks, there's a group of cells, the ASJ and AUA neuron, for example, which appear to branch from some common branch point. And in that case, that actually is what happens in real life. They actually share a common progenitor, and that progenitor is then the sister of a progenitor for another neuron called ASE. You can see that branch as well. So that's all good. But then there's other stuff here that doesn't make sense, right? So for example, the ASE neuron itself actually is really two cells. There's a left and a right. And most of the cells in the embryo are left-right bilaterally symmetric, and we don't resolve them. We basically see, ASH neuron, for example, there's two of those, and we just see them all pooled together as ASH neurons. But for ASE, after it's born, they actually become molecularly distinct, the left and the right version. And the ASE left neuron is actually over here. Like it totally teleported to some randomly different place of this projection. So again, like those of you who work with these projections a lot know that, this is maybe not too unexpected, but this kind of thing happens, and it's something certainly be cautious about over-interpreting proximity in these kinds of diagrams. Right. So just taking another step towards the early annotations, we basically do the same kind of annotation, looking at these different projections for cells that just came from early embryos, and using the imaging data that I showed you before to annotate them. So a schematic of how this works. We have this, various genes whose expression pattern we've mapped by lineaging, like the STAY51, the MS lineage, and then only a subset of the cells that are derived from that lineage express HAND1 or 39 genes. And so we can combine these to get very high resolution pictures of which cells come from which lineages. And this is sort of overwhelming view of sort of what some of that annotation looks like. I just want to highlight this one point from here again about the sort of where the projections can lead you astray. So just zooming in on one small part of this over here, you can see that there's this branching pattern again, and which the tips are correct, right? So the red group and the red cells here and the blue cells, those correspond to the red cells and blue cells up in this schematic of the true lineage on top. But actually the next branch here, these are not sister cells, these are actually cousin cells in the real lineage, and the sisters of these are actually in some other part of the plot. So again, with a high resolution, we're able to tease this apart in C. elegans because we have all of these markers. I think this really emphasizes the value of some of these experimental barcoding and lineage tracing approaches that we've seen in some of the other talks of this session. So at the end of the day, we have the entire lineage essentially, well over 90% of the cells covered by the single cell RNA-seq data. where we can, and the coverage is pretty high. We have hundreds of cells from most of these branches, and so we're able to get pretty high accuracy quantifications of transcripts for all but the most lowly expressed genes. Just an example showing RNA versus protein for this one for gene that we looked at earlier. So so that's great. Like what we learned from doing all this? I'm going to highlight just a couple of examples of things we emphasize in the paper, and then move on to talking about timing for the last few minutes. So one thing is that there's a lot of examples where cells from different lineages converge to the same fate, and actually that's pretty common. And molecularly, we appear to nearly completely lose the lineage history when that happens. So the cells become indistinguishable. You can't see their lineage markers anymore after they come together. There's a lot of examples of what's called multi-lineage priming, where you have a regulator of a particular cell fate that's actually expressed in the parent before that cell is fated, and then it disappears in the sister of the cell where that regulator is not affected. Early on in development, most of the transcriptome changes we see are really highly correlated with lineage identity, but then there's a rapid transition when cells start to differentiate, where often there's a large jump in the UMAPs as well, and the cells switch from the transcriptome changes being much more correlated with their terminal fate. This is just an example of some of these jumps, and we've seen a couple of those already. For example, this IL socket type 3 parent cell, you see one of its daughters is a type 3 cell, which is contiguous, but then the other cell is in a totally different place in the UMAP. And we can actually quantify, not just looking at the UMAP, which can mislead you, but looking at the levels of gene expression differences that have occurred in these different branches. And actually these teleporting branches actually have more quantitatively more changes in gene expression on average. All right, for the last few minutes, I want to talk about how this project has helped us think a little bit about really a fundamental question in embryonic development, which is how you achieve precision in gene expression. And I told you already that it's an incredibly robust process, the same pattern of cell divisions in every animal. So that's going to require spatial precision in terms of expressing regulators in the right cells, temporal precision to express them at the right time, and also dosage precision to express them at the right level. And actually, Arjun's postdoctoral work is sort of like some of the most beautiful work showing how important this can be, where basically the idea is to specify a particular fate, you need to increase the levels of a particular regulator to a particular threshold level so it can induce that fate robustly. And it has to happen during the right time window as well. In the case of C. elegans embryos, it's happening in the context of like a 15-minute cell cycle in some cases. Right, and this precision can come from a lot of different mechanisms, right? So like in the fly embryo, in the blastoderm, and other tissues like the C. elegans germline is another example of this. We take advantage of spatial averaging in syncytial tissues, where individual noisy nuclei can share transcripts across the cytoplasm. In slower developing systems, you can have temporal averaging and just rely on a long time for transcripts to accumulate and achieve some steady state level. One thing I think that can be very widely used and maybe discussed a little bit less often, but is probably the most important mechanism in developmental context is redundancy. That's both things like redundant enhancers, shadow enhancers, or so as they're called sometimes, or even like redundant paralog regulators that basically have the same function and sort of can buffer each other's noise. And so in the C. elegans embryo, and this is what Arjun's work showed, and we got into this actually through a collaboration with Arjun. We got to Air's beautiful 2013 paper, showed that it involved, so Arjun's work involved the robustness of the specification of intestinal fate, and this lineage is called E. And basically, if you look across expression across time for the key regulators of this, there's these genes called N3 and N1 and L2, and N3 and N1 turn on L2, and L2 is the master regulator of gut specification, and they have these thresholds in terms of making that specification. But what I want to highlight is something a little bit different, which is the incredibly high levels that these genes go to. So N3, for example, goes from 0 to about 600 mRNAs in a single cell in about 15 minutes, which is a really impressive rate of transcription. Right. So we looked at the single cell data, and this is actually looking at a different data set than ours that was focused more on the early embryo, but the same basic logic applies actually to the analysis of ours as well, and see similar things. And so basically the idea here is, can we learn something about absolute levels of transcripts from single cell data? Which we know there's biases from sequencing. So is this accurate? So we went ahead and we just calculated what are the number of UMI or number of reads, depending on the data set in a mother cell compared to the daughter cell, and use that to infer how much new transcript has been produced in that one cell division, and use a series of cutoffs to basically find genes that we thought were expressed at exceptionally high levels. You can see that at 4 cell stage, there's very few of these, but by the time you get to 8 or 16 cell stage, you actually have hundreds of genes that are expressed at this level where they're increasing by more than 100 transcripts in these very short cell cycles. And so we did some single molecule RNA fish, again, taking advantage of Arjun's lab and their expertise and their microscopes. And this is some example. You can see like really high spot counts for these genes, like X35 or C51, which are all conserved transcription factors, as well as being expressed at these very high levels. They're involved in different lineages state specification. Right. So overall, if you look at the maximum level measured by RNA fish versus by single cell RNA-seq, the correlation isn't perfect, but it's actually it's very consistent with what we saw. With again, an earlier collaboration with Arjun looking at melanoma cells, where basically you're able to predict the absolute levels probably plus or minus twofold or so. And so we think that's enough to identify these very high rate genes is a pretty accurate proposition, right? So I just want to emphasize, how high these levels are, right? So 2 examples in a particular lineage, TBX35 and C51 is the parent, this is the daughter cell of this MS division, right? And so C51, as an example, goes to almost 1,400 molecules in again, like 20-minute cell cycle. So these are accumulation rates of dozens of transcripts per minute. You're essentially packing the polymerase, almost as close together as you can if you assume in vitro transcription elongation rates. So I'm not going to go through the details of it, but basically Priya Ramakrishnan, who's been doing this work in the lab, has done a lot of dissection of using these listed genes that we identified from the single cell data to identify features of these. This was one example where one of the features she identified is these are high rate genes are enriched for this INR promoter element. And if you mutate this element in the C51 promoter, you can see you decrease expression by almost half. And sort of long story short is that we think that these genes, there's not like one specific mechanism that controls this really high rate, but basically all of these genes have a number of different promoter elements that combine to give you this exceptionally high rate of transcription. And so this is sort of finishing with a model for why we're seeing these really high rates of transcription going on. So what's happening in the C. elegans embryo, you have, I said before, it's like a 15, 20-minute cell cycle. You have to make a decision. And then to become a particular cell fate, cell's going to divide and have to make new decisions. So you really need the protein level for the regulators to have a step function like pattern of change, right? And the way you achieve that is to have something like this for the mRNA, where you have this incredibly high spike that's very transient, and then goes away. And actually, we can see this offset here, going back and looking at the RNA versus protein levels for some of these regulators. So on the left, we have this is doesn't really matter which transcription factor it is, but this is the single cell RNA-seq measured mRNA levels, and then on the right is the protein levels for the same gene as measured by GFP in our imaging studies. We have hundreds of genes we can do these for. We always see the same pattern where there's this lag. You can see if you plot GFP versus or protein versus mRNA, you get a pretty poor correlation, similar to what Shaleb was seeing in the intestine. what's cool here is and we're just starting to do this, now we have the tools to be able to actually not just learn the protein degradation rate and the translation rate, but actually to look for exceptions within particular lineages where it looks like there might be not well fit by a single model and there must be some kind of regulation. So I just want to close there, thanking people in my lab who have done really well, surviving from times we can be close together to times when we can't. Philadelphia is an amazing city. People who are interested in postdoctoral opportunities or in graduate school, highly recommend it. And I'll stop and take questions.
Thank you very much, John. We are a little over on time, but I think we can maybe squeeze in one question or two. So Christian Ray asks, well, first says cool work, and then says, do you have any idea about if tissue homeostasis or lack thereof in C. elegans may affect developmental trajectories or lineages?
Yeah, I'm not sure I totally understand the question. So tissue homeostasis. So what's interesting about C. elegans is like in most cases, there's very little sort of compensation for changes in neighboring cells. So for example, if you ablate a particular cell, the 16 cell state, basically the cell fates that would have come from that cell don't appear anymore. Whereas most of the other cells, they produce their normal fate. So a few exceptions, late state inductions that happen. So yeah, so basically we think that there's little of that. But we're quite interested actually. Then there's interesting ideas we have for single cell approaches to try to test specifically for cell fates that require to basically cell contact in the embryo to be induced. And I think we perhaps will identify more of those if we look systematically.
I think since we're a bit over on time, we will just move on to Katie. But thank you again, John.